Welcome back to the Classics of Immunology Journal Club. And before we get started, don't forget to like the video if you like it, and click on the bell so that you get notified for the next go around. And then also check out the on the YouTube channel there, scroll down and come to my website, subscribe to the channel if you'd like. And also on my website is a writing tab on the home screen. Click on that and you get all the classic papers of immunology. So today we're in 1987. 1989. What am I talking about? We're almost coming into the modern era. And we're going to discuss two papers from a group that was headed up by Craig Thompson and his crew. Craig is now the president of the Sloan Kettering Memorial Institute in New York City. And so this was back in the day. The first paper, Craig Thompson was the first author. And then the next authors was Tullia, Lindsten, Jeffrey Ledbetter, Stephen Kunkel, Howard Young, and Stephen Emerson, Jeffrey Leiden, and Carl June. Title of this paper is CD-28 activation pathway regulates the production of multiple T-Cell-Derived Lymphokines / cytokines. Came out in the Proceedings of the National Academy of Sciences in February of 1989. The background on this paper is that the real story started on this CD-28 molecule that was found back in the early days of the 70s and 80s. As you will recall, actually it even goes back to the 60s. Joe Oppenheim's group showed that if you took away the macrophages and you activated the T cells with something like PHA, that you get a markedly diminished response, if at all. So there was something the macrophages were doing. And early on, that led to one of the things that led to in the early 70s, 1972, there were two papers that came out in the JEM showing that the macrophages secreted a soluble product that was named lymphocyte activating factor. And that subsequently became interleukin-1, as you may recall. So it looked like the macrophages were, at least one of the things that they were doing, were making cytokines that would help the T cells make cytokines and therefore proliferate better. And the cytokines that the T cells were making was interleukin-2. So those were two signals that the macrophages were giving, because the other signal was the antigen, the peptide antigen. And as you recall, we just last couple of weeks discussed the papers by Pamela Bjorkman and Don Wiley and Jack Strominger, where they solved the structure of the MHC encoded molecules that are on the surface of the antigen presenting cells, primarily the macrophages, but also on B cells. And for the first time, it was realized that the antigenic peptide fits into a groove in the MHC molecule on the surface of the macrophage or the B cell that then connects with the T cell antigen receptor. So we have the T cell antigen receptor, we have soluble cytokines that the macrophages are making. But there seemed to be also a third signal that the macrophages were doing that required, in addition to the peptide MHC, in addition to that, there was something else that seemed to require cell-cell contact. And that's the story that we're going to talk about today with this CD-28 molecule. Now, so nothing really else happened until 1980, when people searching for surface molecules on lymphocytes found a monoclonal antibody that was bound to a molecule or molecules on the surface of T cells, but not macrophages or B cells, and it wasn't the T cell receptor or anything associated with that. And then nothing much happened until 1987, when a cDNA clone was obtained that coded for that T cell surface molecule. And by this time, it had been given a designation of CD-28. CD stands for cluster of differentiation molecules, because it was wasn't clear whether or not there was just one molecule in a lot of these situations or whether there was more than one molecule. The cDNA encoded 44,000 Dalton protein that had the domains of the immunoglobulin superfamily. So that was interesting because the antibody molecules and the T-cell receptor by now was known to be in this same family. And it's expressed on it was found to then be expressed on the surface as a dimer, a homodimer of these 44 KDA molecules. So that's the background up until we're getting to this 1989 paper. In the introduction of this paper, the authors say that they showed in 1987 that antibodies, so they used the antibodies to the CD-28 molecule in lymphocyte activation experiments, and they found that they would, if you threw in the antibody in macrophage depleted cultures, that you could markedly increase interleukin-2 production. And the question is, how did that work? They thought, well, maybe it increased the number of cells that were proliferating because they were measuring thymidine incorporation. Or alternatively, they thought, well, maybe we're just increasing the amount of interleukin-2 produced per cell. And so they decided to do a series of experiments that were reported in this paper. So in the materials and methods of this paper, the cells they used were human peripheral blood mononuclear cells. And they selected T cells by removing the macrophages from the PBMCs through plastic adherents on the bottom of culture flasks, which by this time was standard procedure. And the antibodies that they used, the monoclonal antibodies that they had available, were the anti-CD-28 monoclonal antibody and the anti-CD3 monoclonal antibodies. In order to activate the lymphocytes, they used the anti-CD3 because that's the CD3 complex is the signaling complex of the T-cell receptor. And they use that as an adherent layer in the bottom of microplates. And then they used the anti-CD28 molecule that was soluble in suspension. And then they would harvest the cultures at different time intervals after they activated them with anti-CD3 and anti-CD28 and so forth. And they would then extract the RNA from these, total RNA from these cells, and they would then run these out on gels and do northern blots using a radiolabeled cDNA probe that was specific for the different cytokines that they were interested in. And so that they would mix it with the radiolabeled cDNA probe. The cDNA then would hybridize to the messenger RNA for these different cytokines. And they wanted to see whether or not this anti-CD28 would actually increase or whatever it did to the message. So they had cDNA probes by this time for interleukin-2, for interferon gamma, interleukin-4, TNF alpha, and lymphotoxin. They also used a negative control, an HLA molecule, and they also had available the Lymphokine assays that I won't go into because we've gone into them in detail. We need to screen share now. This is figure 2 from this paper, and these are northern blots. And up here is the total RNA, and it's a poor reproduction, but it was exactly the same amount of total RNA in each lane. And here's we have medium, so that in this lane, and then we have anti-CD3 alone after 6 hours after the activation step, 12 hours and 24 hours, and then we do the same thing with anti-CD3 plus anti-CD28 at 6, 12, and 24 hours. So you see what's going on here. It's very interesting. Here's interleukin-2 is clearly positive at 6 hours, but then negative at 12 and 24 hours after activation. But if you add in anti-CD28, first of all, you see a marked increase in the messenger RNA at 6 hours, but you also have clearly evident RNA at 12 and 24 hours. The same general pattern, although not as impressive, with TNF alpha, increases when you add, the message increases when you add CD28, and it persists in the same way that IL-2 did. Lymphotoxin is not as impressive here. There is an increase with anti-CD28. Same way with interferon gamma. And here is GM-CSF, and you can see a faint band here at 12 hours with interferon gamma, and it's a little bit more easily discerned here with GM-CSF. And 4F2, which is a membrane molecule that was known at the time and subsequently was found to be an amino acid transporter, it doesn't really change a whole lot with plus minus anti-CD28. And of course, here's the HLA antigen, and the message doesn't change at all. So that's all I want to show you from this paper. We should stop this share and go on then. And they quantified the amount of RNA that was increased by the anti-CD28, and it went up in these various cytokines anywhere from 5 to 50-fold, which was a huge amount. So in the discussion of this paper, they concluded that there is a CD28 signaling pathway that's activated by the antibody to CD28, and it functions to increase the expression and secretion of acetylymphokine genes, and it seemed to be what it's raison d'etre, its purpose in life. The one thing they didn't see was an increase in interleukin-4 messenger RNAs or protein. But they concluded, or they thought then, they speculated that the CD28 signal may regulate the transcription or the transition of interleukin production from an autocrine to a paracrine level, so that there's so much more cytokine around that it could influence neighboring cells, which is an interesting concept that I don't think anybody really has followed up on. But the other thing they speculated on was that this may be possible to increase in vivo immune responses pharmacologically by altering the CD28 signaling pathway in some way. And of course, that made them, then they also wanted to know, so they wondered whether or not there was a ligand on the antigen presenting cells, macrophages and the B cells, that would activate this CD28 pathway. The very same group, very same year, 1989, in April, the first paper was out in February. In April, there's another paper, but now the authors have been scrambled. And now we have Tullia Lindsten as the first author. Then with Carl June, Jeffrey Ledbetter, Gregory Stella, and Craig Thompson on this paper. Title of this paper is The Regulation of Lymphokine Messenger RNA Stability by a Surface-Mediated Activation Pathway. This paper got out in Science in April of 1989. So, you have to keep trying. Now, the interesting thing to me about this paper, which I didn't know really until I looked up all these authors and made sure I knew where they came from and so forth. Tullia Lindsten was married, I think still is married, to Craig Thompson. And she came from the Karolinska Institute with an MD, PhD. So she was no slouch, essentially. And basically, they've worked together their entire careers. So I think that was a good deal for both of them. And this was when Craig was at and Tullia were at the University of Michigan at that time. Carl June was an old friend of Craig Thompson's from the Bethesda Naval Hospital. And Jeffrey Ledbetter was with the Oncogene Corporation in Seattle. In the introduction of this paper, they pointed out that they've done their homework, and they pointed out in bacterial systems, there were both short-lived RNAs, messenger RNAs, as well as much longer-lived messenger RNAs. And the short-lived RNAs seem to be the ones that were associated with the responses on the part of the bacteria to growth conditions, particularly when they got nutrients and so forth so that they could proliferate rapidly. The messenger RNAs would be the short-lived messenger RNAs were the ones that were responding. And they pointed out that in eukaryotes, There was a similar kind of a phenomenon. Messenger RNAs associated with cell growth, as well as proto-oncogenes, and those associated with DNA replication were all short-lived messages. The other thing that had been shown in the molecular biology literature is that if you blocked protein synthesis in your different cultures and so forth with cyclohexamide as a drug, that it would stabilize messenger RNAs, the short-lived ones, and so that they would last longer. And so it was conjectured, speculated, that there might be a cellular RNA degradation system that was proteinaceous in nature, and that cyclohexamide would block something in that pathway so that the messages could last longer, or did last longer. It was also speculated by these same people that cytokine regulated genes could be regulated, the cytokines, by the stabilization or not of the message. And it looked as though when they looked at the sequence of the nucleotides in these messages, that in the 3' untranslated region of many of the cytokine genes, as well as the oncogenes and so forth, there were stretches of poly-AUs, and you needed to have at least 3 different stretches. They lasted anywhere from like 8 to 10 nucleotides to maybe even up to 20 nucleotides of these poly-AUs in there. And they wondered whether or not that this was the recognition site on short-lived messenger RNAs that were going to be targeted for degradation. Stranger things have happened. We should go to essentially the same methods that were used in this kind of a paper that were in the first paper. So let's go to the screen sharing once again. So this is the second paper where Tullia Lindsten is the first author. And this is figure 4. And this shows the rate of messenger RNA degradation in T cells stimulated with anti-CD3 plus anti-CD28 at 5 hours. At 5 hours after activation. At that time point, they added actinomycin D, which is a drug that stops transcription. So if you have any message that's present at this particular point in time, you can follow its degradation rate over time. And what we have, so that we've got arbitrary amounts of message at time 0, which is 5 hours after activation. It's the time that the actinomycin D was added. And I want to get my notes so I don't get you more confused than I am. And then we've got to scroll down a little bit so that I can show you the time interval. So this is time after actinomycin D. So we go 15 minutes, 45 minutes, 90 minutes. And so these are pretty short-lived messages. And you can see what happens is when you add, these are the symbols here, TNF alpha and GM-CSF. Up here is plus anti-CD28, and down here is without anti-CD28. And you can see that that these messages really are degraded pretty rapidly, but for both TNF and GM-CSF. But when you add in the antibody to CD28, they're basically very, very stable. Same thing with interferon gamma and IL-2. And so these are the plus CD28, and this is without CD28. Down here we have, and this is c-Myc, and it didn't seem to make any difference whether you had CD28 or not. And this is the 4F2, the amino acid transporter. It's stable with or without anti-CD28. So those are the kinds of data that they generated in this paper. And they were pretty clear cut. So in the discussion of this paper, they concluded that anti-CD28 stimulation by the antibody stabilizes cytokine messenger RNAs. The AU-rich sequences seem to be the really important things, and that was borne out later on in other experiments by other people, actually. And so that was it. And that was the reason it made science and so forth, was because nobody had an inkling that this is how some of these cytokine genes might be regulated. So subsequently, the beat goes on. This was 1989. Also in 1989, Art Weiss, who was at UCSF in San Francisco, did experiments looking at the effect of anti-CD28 on the transcription of the gene of the different cytokine genes, and showed basically, particularly with the IL-2 gene as the prototypic cytokine, that if you triggered anti-CD28 at the same time, you had a 5-fold increase in the transcription rate. So that means that there was both a transcriptional mechanism and a post-transcriptional mechanism that anti-CD28 regulated that resulted in more interleukin-2 production, and then also these other cytokines as well. Then in 1993, now you see we're going on now from 1980 when the original antibody of the anti-CD28 was generated, and now we're up to 13 years later to 1993. And Howard Young at the NCI in Frederick showed that the CD28 transcriptional response element, the stretch of DNA upstream of the IL-2 gene, was a REL site, REL, which meant that it was a NF-kappa B site, which is a more popular designation of the whole REL family of proteins. That was interesting because REL had not been, up until this time, hadn't been really NF-kappa B thought to be involved in the transcription of IL-2. And then in two very elegant papers in the mid-90s by Ellen Rothenberg at Caltech, showed in 94 and also another one in 96, was that there were 3 families of transcription factors that had to be co-expressed and active in order to get IL-2 gene transcription. And if you took any one of them away, and those 3 were AP1s, an AP1 site, a nuclear factor of activated T cells, and NFAT, and then the REL site. If you took any one of those 3 away, then transcription would shut down. And of course, cyclosporin A was found to inhibit NFAT generation, and glucocorticoids was shown to inhibit the REL site. And so that's how these things were really figured out. And then Shochi Liu, who was a colleague of mine at Cornell, showed in 1999, so we're almost 20 years later, that the REL had several family members, at least 3. And she was an expert in CREL, which was the cellular proto-oncogene of NF-kappa B, REL. That was not expressed in resting T cells. And so the reason you could get them activated to make IL-2 is that there was a REL family member, REL A, I believe, that was already in the cytoplasm and ready to go. So the initial activation via anti-CD3 and anti-CD28 would activate REL A, and then REL A activated CREL. And CREL was a very, very vital component to get sustained activation of the IL-2 gene after T cell receptor and CD28 stimulation. So there's a lot of immunology that went on in these 20 years between 1980 and 2000. And if you want to know more about all this business, what you should do, I have a solution for you. You should read my book. My second book is called Molecular Immunity, and it's a chronology of 60 years of discovery in immunology, essentially. So you can get that online. Just go on Amazon, and you can even get it as an ebook if you want. And the chapter that really deals with these, so this became to be known as co-stimulation. Co-stimulation, the stimulation was really thought to be the anti-CD3 and the peptide MHC antigen activation of cells. And so CD-28 came to be called the second signal of T cell activation by these guys that were primarily interested in T cell receptor activation. They sort of ignored the cytokines, the pro-inflammatory cytokines, because it was not just interleukin-1, it was also TNF and interleukin-6 that would act as co-stimulators of T cell activation. And of course, they ignored the other, and so there were at least three signals that the antigen presenting cells were giving to the T cells: T cell receptor, CD-28, and cytokine and pro-inflammatory lymphocyte activating cytokines. So, but in order to really get proliferation, they all had to work through IL-2. If you blocked IL-2 gene expression with, say, steroids to block REL, you wouldn't get any proliferation. Or if you use cyclosporin A, and that's why it works in transplant rejection and so forth. So basically, there's four signals that are necessary for T cell activation, and this chapter in my book, and it also goes into the ligand for CD-28, which are a family of molecules called B7s that are on the surface of the antigen presenting cells. And then, the beat goes on, because what was discovered subsequently, after all of the co-stimulators were discovered, were co-inhibitors. So the famous co-inhibitors are CTLA4 and PD-1. And that is also discussed in this chapter in molecular immunity. So happy reading. Don't forget to like the video and subscribe to the channel and do all of this stuff. It'll get you going, and you'll really be learning immunology. So thanks for listening.